So here's a different idea to approach DNA sequencing with. So you take these reads that may be, say, 100 letters long, and you break them up into overlapping pieces, say 25 letters long. This is a parameter you can adjust, but say 25. And the first 25 letters, then base 2 to base 26, and so on. So you have all these overlapping guys. And you do that for your entire project, all these millions of reads. You merge the 25-letter guys that are the same, and then you look for something called Eulerian paths, which I'll tell you about in a minute. Let's see. Yeah, let me illustrate this. So we're going to build an Eulerian graph over this short sequence of DNA using k equals 3 instead of 25. 3 is much more manageable. So our first triple is ATG. And ATG is the invisible guy up there. We've gone from the first two letters AT to the next, to the terminal two letters here. So if we were doing this with 25, it would be, each of these would be 24 letters long, and the guy in the middle would be 25. And then we keep doing this. TGT, and there's a reason I'm going uphill here, then so on, and we fill out the rest of the graph, just going through here and doing this overlap thing using triples up here. So that's our graph. Now, what I described we're going to do with the sequencing project is we're really going to start with the graph and try to figure out what the sequence is. How hard is that? And if you look at this just trivially simple example, it looks like I have to be a genius to know at this point to go up here and so on when I have decisions to make. Well, let's go across it and let's just do this ourselves. So the first step, we start here with AT and we add a G to it, so that gives this G here. And now we've got a decision to make, and you think we'd want to go up here, but let's just imagine we go horizontally, because we're just following these arrows we don't know. So when we go horizontally, we just remember there was an unexplored path back there. We have to go back and find it. Do the same thing next. Now what do? Well, we had two places that we didn't explore. This path, for example. So we go back and take it, and then we follow it around. Come back where we were, so we've inserted these missing letters into our sequence. We don't have to be smart to do that, we just have to remember where we didn't explore that. And the same thing back here, and we've deduced the sequence from the graph. If we did this, the Hamiltonian pathway, that is to take all these triples and put them on the nodes of the vertices, the graph is much more complicated. And we do have to be smarter to figure out a Hamiltonian path that would visit all of the vertices once and only once. And the guy we just had is A, simpler, and B, we don't have to be smart. This was something that my postdoc and I published this very new algorithm in 1995, and absolutely no one paid any attention to it until the early 2000s when lots and lots of data was being produced, and they went, oh yeah, we got to work on this. So it's not the only thing I've done that people ignored, but it was one of the few that they went back and said, that was a good idea. But we, at that time, Ramana and I, we realized that we were asking for a lot of memory. But we also didn't want to give up any information, so every k-tuple would have affixed to it the read it came from and the position in the read. So it might have a long list or a short list. So this is the soup. Take each read, break the read into overlapping k-words, merge the identical k-words, and find these Eulerian paths. Hope I have this somewhere. One of the big problems with sequence assembly always was knowing there's two different genomes you're trying to sequence. One is the complement of the other one. And you never know which the read comes from one way or the other, and you need to get that consistently. So that's a real problem. You have 27 million reads, you have 2 to the 27 million different concatenations of this. And Ramana and I realized that we would double the computation time if we took each read and put in the reverse complement, but we would eliminate all of this orientation stuff. So that was part of our suggestion. But we're really asking for even more memory than this does. So again, just a picture of this, I guess. Here's the blue read, and it goes into its blue reads. It overlaps the purple read here, and so on and so on. So what you're trying to do is reduce this highly complicated problem to something that's much more one-dimensional. And if all of these keywords are unique in the genome, then it's linear time. So you have a linear time approach to genome sequencing, which is pretty good. There's no pairwise alignment, which I made my living doing for a long time. There's no layout, there's no consensus, and one-letter insertions and deletions, which drive us crazy in sequence alignment, are really not any more problem than the letters that are opposite one another. Orientation, I just mentioned, these consistent orientations, we just doubled that. So the problem, and of course, every new method has all kinds of problems. The problems here, of course, there are edges that are wrong. The graph is tangled. Remember all those ALU repeats that are filling up our genome. And the storage requirements are amazing. Ewan Birney at the European Bioinformatics Institute I think his grants were mostly more and more storage as he was pursuing this. So what's this business about the edges? If you have an error here, as the tuple is going over that error, there'll be pieces that are, or 25 letter pieces that are not correct. And if you view that as a bubble, I don't know why this is green, but there it is, these errors off here. The expected depth of coverage of one base in this next generation sequencing can be from 30 to 100. So it's hugely deep. In the Human Genome Project, 3 to 5 was the depth of coverage. So the depth is huge. So if you have a graph, and what Ramana and I did was just trim these guys off that were lightweights. It didn't occur very often. What happens now in essentially all of the packages that do this is they try to change that letter back so it would be consistent. And they call it error correction, which you can call it what you want, but that's another difference. And a kind of classy statement of this problem due to Pavel Pevsner. In this graph, this is our K-tuple graph for the genome, and the colored guys with arrows on them are reads. And you want an Eulerian path through this tangle that breaks as few reads as possible. That's the idea.